I welcome you all to today's event, which is the newest edition of our series of talks called EV Fundamental Biology. EV Fundamental Biology is dedicated to uncovering the core mechanisms of extracellular vesicle formation, function, and communication, as well as the experimental technologies that allow us to observe and interrogate the behavior in living systems. Today, we are honored to be joined by Dr. Charles Sly, who will be delivering a talk titled Seeing is Believing, or is it? EV Imaging and In Vivo Visualization. Dr. Charles Sly is a research fellow at the Institute of Atomic and Molecular Sciences, Academia Sinica, Taiwan. His work focuses on creating molecular bioimaging tools with high spatiotemporal precision to interrogate biological systems, notably bionanoparticles like extracellular vesicles and DNA repair. His cutting edge bioimaging platforms are used by more than 65 laboratories worldwide, fundamentally advancing research in cancer biology, bioengineering, and therapeutic development. His publications appear in leading journals, including ACS Nano, Advanced Materials, Advanced Science, Nature Communications, Nature Protocols, and Nucleic Acids Research. Today, Dr. Lai will present work his lab has led for the imaging and visualization of extracellular vesicles in vivo. I request the audience to put their questions in the chat box, or you can just unmute and ask the question at the end of the talk. Now, Professor Lai, the stage is all yours.
Thank you, Ayush, as well as Bárbara, for co-moderating my talk today. And I'm very honored to be invited by SNAF to give or to contribute to this EV Fundamental Biology series. So before further ado, let's go on. So I guess I made the title quite tantalizing as to, are you sure that what you're seeing is believing? And let's head into this field. Let me proceed to the next slide. So this talk is pretty much a collection of most of my education talk in the past 2 years at the ISEF annual meetings. So it's sort of like I used to have the, what do you call it, extended play, the EP of an album. And now this is sort of like an LP, the long play that I try to put together. And I kind of remaster, rearrange, and I hope it is okay. It's not meant to be sophisticated, so please don't mind me if I kind of dumb it down or simplify it. It's really meant for newcomers to have a general sense of what is going on when you're trying to image vesicles. So I guess to echo the titles, seeing EV is believing. And because I am MIT, made in Taiwan, but grew up in Canada when I was younger, it's pretty funny is that if you hear Canadian bacon, usually what comes to your mind. And when I first got to the States or in Boston to do my postdoctoral work, at least that's what my American friend usually joke with me is like, hey, Charles, where are your Canadian bacons? And of course, most people would think that this is what Canadian bacon look like. And lo and behold, Canadian bacons are actually ham and they are not bacon per se. So I try to use this as a joke that it is not always true that what you see is what they actually are. So seeing EVs believing, in my opinion, is usually true only when your experiments are properly planned. Now, something about EV imaging and labeling is that, to my knowledge, I think it's okay to say that there's no one perfect method, or at least not yet. And with every method that you choose, each of them requires thorough characterization and or validation to prove that what you're observing is to the most possible true as possible. So I like to watch a movie. I'm sure most of you have watched the movie Life of Pi. And when I was given the opportunity to give a talk at ISEP, I thought, well, Life of EV, Pi, EV, they're all kind of circular or assumed circular. So if you go from EV, chances are people will be interested as to how they are distributed in the body, whether you inject it or took it in, or whether they are endogenously released. A lot of us are interested as to how these little nanoparticles work in the body or distribute in the body. Then once they reach the site of interest, then we may be interested in their uptakes. And then you'll be interested about their fate. And finally, you're hoping that after all this journey, Life of Pi, that there is a biological effect. And hence, I think that's why most of us are in this field. Now, just want to go over being in the field for about 15 years. This is usually the conventional vesicle study flow that you go from dosage to function such that you administer sufficient dosage to see a function. Only then will we be interested in tracking them. Then, if you're really into in vivo, then would you study the biodistribution? And this has been the majority of the flow in the past about 20 years, but it's slowly changing. I'm hoping that by the end of today's talk, I can suggest other elements that people can consider as they plan their project. So why vesicle imaging, especially when they're suggested to carry a function? Well, importantly is that vesicle imaging allows simultaneous monitoring of the spatial as well as temporal resolution of the EVs. And beyond that, you can potentially observe their morphology and their physiological characteristic all at the same time. Further, you can actually observe at different resolution. So you can observe at cellular resolution up to tissue, organ, and whole organism based on the question that you're trying to ask. Importantly, you don't have to disrupt the model that you're trying to study. They're usually non or minimally invasive, meaning that you don't have to fix them or sacrifice them. And in addition, if you do it right, you could potentially at least semi-quantify the vesicle, which is pretty critical in any type of vesicle-related science. And another benefit is that for vesicle imaging, you can avoid some of the sample processing artifacts. We all isolate vesicle if you're in this field. You know that there is also not a perfect method to isolate vesicles. Each method comes with pros and cons. The same goes with vesicle imaging. And then if you do it right, then you can at least bypass some of the artifacts from isolation of the vesicles. Further, you can avoid specimen modification or degradation of the sample or the model that you're working on. And finally, because you image them usually live, you can minimize or reduce sampling error. To go further, vesicle imaging is quite important for both basic biology as well as clinical translation, especially for preclinical studies. If you look at this chart from, there's a book called Molecular Imaging, second edition is kind of old, that vesicle imaging contributes to most of the preclinical work that ranges from target identification, assay development, lead optimization, and all the way to early clinical evaluation. And to be able to image them, track them, really help the preclinical part of the vesicle studies. So I'm kind of old. There's a book called 6 Habits of Highly Effective People or something like that. So and I think later the family published another one. It's called The 7th Habit of Highly Effective People. So I kind of try to break it down to some recommended strategy for vesicle imaging. So the first one is, it sounds obvious, is to identify your question. Yeah, it's really important to identify the question you're trying to ask. And that could range all the way from development to disease, devices that you're trying to make, biomarker, or to use vesicles as a therapeutic vehicle. And after you identify the question you're trying to ask, then I would suggest that you think about the model you're trying to study your questions in. Now, if you look at this picture, it ranges from 1 millimeter from C. elegans to zebrafish to small animals such as mouse, and all the way to non-human primate animals such as macaque. And if you put this into perspective, you're trying to study nano-sized vesicles, then you have to think about the subsequent question that I'll raise, which is the spatial and temporal resolution that you can achieve, and how they can be matched to answer the questions that you just raised in the last part. And this is a cartoon that I usually present. So this is how we think vesicles work. That vesicles can be released by the fusion of multifaceted body to release, say, exosomes, or through the shedding from the plasma membrane to release, say, ectosomes or microvesicles, or through other mechanisms to release nanoparticles such as exomeres, suprameres. All of these carry bioactive cargo, including nucleic acids, proteins, and lipids. Then they can be delivered to the recipient cells and offload these cargo to facilitate cellular communication. And here's a question: what are you trying to study? Are you trying to study both the cargo and the vesicles, or vesicles alone? And those questions should be identified as early as possible when you're planning your experiments. And moving forward, then you want to think about the time and the space that you will be needing to answer those questions from the last 3 parts. Essentially, question, the model, and the entity that you're trying to look at. And something to keep in mind is about your specificity. Like how do you actually label them or not label them and detect them? This is really critical as I'll go through in the following pages. In terms of accuracy, think about the spatial resolution that you need. This largely depends on whether you're tracking the whole animal or you're just tracking them at a cellular level. In terms of the time, it could be really critical if you're trying to witness, say, for example, the docking or the uptake of the vesicles. In terms of resolution, as I mentioned, this ranges. You can go all the way as a whole organism or into subcellular resolution. And finally, I think a rule of thumb is to think about signal to noise ratio. There is no one best method. I think the best that we can do is that after you made a lot of the controls, you're trying to ensure that what you're witnessing is a real positive signal, especially over time and over space. And strategy number 5 is to determine a method after you have gone through the past 4 strategies. So I think the more common one to detect the physical attributes would be, say, DLS, NTA. I won't go into detail. I'm sure there are other experts that can explain this much better than I. ZFU, ISON, just to mention a few. In terms of microscopy, there is the conventional electron microscopy ranging from SEM to TEM. There are variations of it. And further, there is also probe scanning microscopy, the atomic force microscopy that you can use to detect the morphology and the stiffness of the vesicles. Yeah, if you're trying to image or track vesicles, typically you would go into optical microscopy. That may involve fluorescence as well as chemiluminescence and bioluminescence. And further, for the more peripheral study, people have been using MRI and other SPEC or CT methods, or essentially nuclear medicine ways to detect the vesicles. And for the categories that are listed here, from optical microscopy to, say, the nuclear medicine ones, we would generally categorize them under the category of molecular imaging. And this would be what we'll be covering mostly for the rest of today's talk. Now, finally, after you have gone through the past 5 stages, you want to think about how to label them. There are a couple categories. Essentially, the first one is usually from EV-enriched protein, and they could be labeled by antibody or aptamers. And also, you can design reporter to report those EV-enriched protein. And I think the most common one people know would be CV63, molecularly conjugated GFP. Another route to take is a general labeling method, such that you can use, say, lipophilic dyes, or you can use click chemistry to label the vesicle membrane. Or similarly, you can also use reporter to label the membrane of the vesicles. Or you can use lipophilic radionucleotides or contrast agents to reveal the vesicles. Now, just to go over a few considerations to take when you're trying to label your vesicles, I think the most obvious way to think about is location. Do you want to label the luminal side of the vesicle, or do you want to label the transmembrane, the inner membrane, the outer membrane? And all of these have their pros and cons because it might affect the cargo content. And at the same time, if you label on the membrane, would it affect, say, the transmembrane or the outer membrane or the inner membrane dynamics? And at the same time, people have been saying that vesicles can carry organotropism. So if you label the outside, the middle, or the inside, would it affect the ligand protein interaction as they're being trafficked to the different recipient cells? So those are things that you can think about when you're planning your experiment. And this is just to give you some pros and cons of each type of vesicle labeling. The first type would be antibody or slash aptamer-based vesicle labeling. The pros is that you can typically have more specific vesicle population label with what you're labeling with. The cons for that would be you want to have antibody slash aptamer specificity, that you want to make sure that they're really what you think they're targeting. And the labeling procedure may be quite bothersome, or it could be laborious, depending on how clean it is. So you might need to spend a lot of time to clean up whatever does not label onto the vesicles. And it definitely require a lot of controls to ensure that you label them correctly and efficiently. And some examples for these are, of course, antibodies or aptamers against CD63 or 81. So this is not meant to be exhaustive, but just to give you an example of the typical ones that you see in the literature. And another family of labeling is based on reporters. Now, they can be general or specific vesicle population labeling. And they typically do not require staining after you have expressed these reporters into the donor cells and subsequently the release vesicles. The cons for that is, yes, you will have to do some microcloning and you will require the reporter expression from the donor cells. If they don't express well, chances are they won't be labeled as efficiently into the release vesicles. At the same time, you need to do controls to ensure that they are properly labeled. You'll hear this a lot. I'll keep mentioning controls and controls because really keep in your mind, we are always challenged by our reviewers that they do provide the proper controls to ensure that your labeling is correct. So do that at all time when you're planning your experiment. And the third family of labeling is radionuclide or contrast agent labeling. They're great because they typically have very, very good tissue depth or penetration in terms of the labeled signal. The cons for that is, again, it's less accessible to the scientific community to get those radionuclide or contrast agents. Typically, you would need to be stationed close to a nuclear medicine lab. And again, they're typically more expensive than the ones I mentioned earlier. And then again, you do require control to, again, convince your fellow scientists that you're actually looking at label vesicles rather than, say, contrast agent or dye residues. Some examples of this are, say, the technician HMPAO, Indian oxen, sorry, I don't know how to pronounce this, but ZR. And these are typically for SPECT, and for super paramagnetic iron oxide NP, you can use it for other nuclear medicine imaging. And moving forward, I think this might be one of the most common ways of how people label their vesicles through dyes. They're pretty good in terms of providing robust signal, and I think it's very common and popular because they're relatively easy to apply. But they also have their cons, is that if you want to label it quote unquote clean, the washing procedure could be quite intensive. And again, they require a lot of control. And some example of that is lipophilic dyes such as PKH, CellView, Dye-I. There are others such as Membrite. Calcium AM is another type where the dye itself can actually penetrate into the cells, and once it enters, the luminal esterase will actually cleave off the AM part to let the dye remain inside the vesicles. And the other one is carboxyfluorescein succinyl esterase. And this one is also quite good from what I read on the paper, such that it works kind of similar to calcium AM, but in this case, it won't provide fluorescence until it enter into the luminal side of the vesicle or cells. And it will only fluoresce after its active group is cleaved by the esterase within. So from what I read, is relatively cleaner than the other dyes. But then you be your own judgment in choosing what works for you. And I want to just emphasize a little bit more about labeling, and that would be in terms of specificity versus the half-life versus the efficiency. I think these three components are essential when you're thinking about labeling your vesicles. So let's take the lipophilic dyes, for example. This was sold by Sigma Aldrich, and they were initially used to label immune cells for long-term tracking. And if you look at their brochure, the half-life ranges from a week all the way to 100 days. And on the right side is the image that usually shows like your vesicles are like your Easter egg, right? I know Thanksgiving is coming up, so I thought this was matching again. And then if you're trying to label your vesicle with the dyes, and if you have done these activities before, you do know that you all have to rid of the dyes, right? The same for vesicles, you need to wash them off. And not only that, because a lot of the dyes, and this might include contrast agents such as the technician HMPAO, they are lipophilic. They might actually form micelles, and micelles are nano-sized, so they might actually become a false positive signal if you're not careful. And as I mentioned, you have to get rid of the dye, right? How do you get rid of them? There are many ways. You can use ultrasonification wash, you can use molecular weight cutoff, such and such. But those are things that you need to keep in mind, and then to make sure that you're actually getting rid of the dye but not your vesicles. And something that people don't typically mention is the labeling efficiency of the vesicles. And I'm not saying that everybody should have 100% labeling efficiency. I'm just saying that keep that in mind. For example, if you have 100 vesicles and you use your labeling method, Do you think that method is labeling only 10, 20% of your vesicle population? Or are you labeling 80 or even 100% of your vesicle population? And would that affect the study that you're working on? So those are things to keep in mind. And to move forward, you saw this cartoon before, but one thing to keep in mind is about the distance that you're trying to image your vesicles. And I superficially categorize them into short versus long distance. And what do I mean by that? So by short distance, I meant if you're looking at tissue level, for example, you're looking at probably 100 micron range. And for cells, you may be looking at 10 micron. And further, if you want to look, deeper in, you can look at subcellular, and like such as organelle, cytoskeleton, as to how the vesicles are processed after they have been taken up. Yeah, then you're looking at less than 1 micron distance. This I would categorize them as short distances. And they typically yield a better spatial resolution as you use optical microscopy. And for long distances, I would say if you're looking at a whole organism in vivo, that you will range, depending on your study model, it will range from about 1 millimeter all the way to 60 centimeter, such as a macaque. And then if you're trying to look at organ in under ex vivo environment, then you might be tracking somewhere between 10 micron all the way to more than at least 1 centimeter. And they typically yield lower spatial resolution. So there is a trade-off. And beyond that, you have to think about the temporal resolution when you're tracking them. And then if you just think about it from a distance perspective, generally speaking, long distance tracking have lower temporal resolution. We're talking about minutes. And for shorter distances, you might have better temporal resolution. Then that would range somewhere from nanoseconds to seconds. And if you think from a study model perspective, the larger models would have lower temporal resolution, and vice versa for the smaller models. For dimension, if you're trying to do 3D, they're typically much more limited in tracking. Yeah, you typically have to fix or sacrifice your sample in order to do 3D. It is not impossible, it is that there's a trade-off between space and time that you need to think about. And this is just one example that we did before where we did very simple super resolution fluorescence microscopy that you can actually see the exchange of vesicles between two cell lines. One we label with green and one we label with red. And in terms of 2D, the temporal resolution is usually way better. As I mentioned, you can have it from nanosecond to second. Right. And this is one of the earlier work that we did back in Mass General Hospital through collaboration with Paul Torsten's lab, that we were able to create a live window with two-photon microscopy to track the release of vesicles from tumor in animals. Okay, so moving forward, let's track some vesicles. And let's begin with from short distance to long distance. And this would be the in vivo visualization part, in vivo visualization part of today's talk. And finally, there are ways where we can track both short and long distance at the same time. So let's start with the short distance. So I'm going to mention some examples. They're, again, not meant to be exhaustive. I just try to pick ones to represent. So please forgive me if I did not cover some of the really good works. So this is one example using calcium exosome. You can see that this is as early as 2003, where, as I mentioned, calcium is membrane penetrable, and they can actually enter, say, vesicle in this case. And once the esterase activity within the lumen cleaves off the AM tail, then you can actually see the signal come off. And another one is PKH26, where people know that they can label through the lipophilic dye, and then they can track where the vesicles are going and how they are taken up by the cells. Okay, you heard something. Is there a question? Okay, I guess I'll continue. Then another one is carboxyfluorescein succinate middle ester. And then this one, in this case, was by Escrambante. This was first published in 2011, that they labeled the vesicle in such a manner, and they were observing the uptake of the vesicles by SKOV3 cells. And they also co-label with tubulin to track how they may actually migrate along tubulin once they've been taken up by the cells. So those were the dyes. And then for the reporters, CD63 GFP, I think this is one of the very first CD63 GFP reporter study that I could find, where again, like because everybody knows that CD63 is an enriched protein in vesicles. So they conjugated or molecularly conjugated to GFP to track the vesicles released by the cells. And another one is a work that I did during my postdoctoral time is that we use a palmitoylation moiety to label either GFP or TD tomato to the inner membrane of the lipid bilayer of the cells. And once you do that, you can see not only the cells membrane being labeled, but you can also track the vesicles released by them. And another one is tetraspanin 4 GFP, where they were able to, I believe this is from Ma et al. From the Ayush group from Tsinghua University in China, where they were able to use this reporter to track microsome release, like pomegranate-like structure. And it looks really unique. And another one is pH lowering, CD63. This is essentially pH sensitive GFP protein, molecularly conjugated to CD63. And this is really cool because since it's pH sensitive, the color will change once it has been taken up by the cells. And this is from Elisa Weaver's group. And they actually have two versions. I think the second version they claim to be better. So it's worth checking it out in their 2020 paper. So, and there's another one that actually can track cargo release. There are a few others, but I really like this one. This was by Joshi et al., and it was published in ACES Nano in 2020, where they can actually track cargo release from the vesicles. So I'm really abbreviating this excellent work such that they label the vesicles with CD63 GFP, but at the same time, they have the recipient cells expressing this anti-GFP forward body. So essentially, if the vesicle releases the cargo into the lumen of the cells, it will be picked up by the anti-GFP forward body to detect them. And kudos to them, they actually did correlative microscopy to not only track them under fluorescence, but also witness the release of it on their EM. So this is a pretty cool paper, in my opinion, to show that the vesicles can effectively release cargo. And continuing on, we have gone over short distance, and let's go into some long distance work that people have done. But before we go into that, once we go into in vivo, essentially we're trying to study vesicle biodistribution, right? And it's pretty important that you are able to quantify them in vivo. And I think, sorry, I'm just trying to make it cute that I brought my daughter to the zoo and I saw capybara, and I was just like, wow, they are really cute animals. Do scientists actually use them as a study model? And in fact, yeah, scientists do say that they are a very suitable model to study. So yeah, scientists are not always benign. But in any case, essentially, like you're trying to study how vesicles are distributed in the body and how they are cleared out. And this actually resonates with what we discussed before, is the time, the space, and how they move about in the body. And at the same time, you have to think about a resolution that you would need to track them in vivo. And so again, this ranges from tissue to cellular to subcellular resolution. Again, this goes back to what question you're trying to answer. And this is quite important, in my opinion, because if you look at vesicle study in the first 10 years or so, a lot of us focus on the origin, the composition, and production of vesicles. And then essentially, then they go into pharmacodynamics as to how these vesicles impart an effect to the treated samples or the treated animals. You don't see as much work done on the pharmacokinetics of vesicles. Essentially, this goes into biodistribution, the uptake, and all that. And personally, I think this part is quite important in our effort of translating vesicle clinically. And further, we're trying to explain how they work, right? If you don't know where they're going, if you don't know how fast they are cleared out, then you lose a lot of important properties in trying to explain how vesicles may work in organisms. This not only apply to therapeutics, but this will go into basic biology as well. And I think focusing on the biodistribution and uptake in study models is quite important and it's worth the effort to go into. And further, vesicles are not always non-harmful. Vesicles might actually be not good for whatever you're treating them with. So this might actually go into vesicle toxicology that people can entertain their thoughts on. All right. So for vesicle distribution, again, I superficially categorize them by two categories. One is exogenous vesicle. This typically will involve administration of a vesicle into your study model. And this could be, say, for example, intravenous, intraperitoneal, intramuscular, skin. It depends on how you administer them. Then you would think about the dosage that you're applying. Reason why I mention about this is that dosage is really important. More is not always better, as you would know for every drug, any type of drug. And I believe the same would go for vesicle. So again, dosage is something to consider when you're thinking about vesicle biodistribution experiments. Another category would be endogenous vesicle, such that are you interested in study specific tissue-derived vesicles? And how long would they last, or how long would it be circulated? So these two categories are both interesting, but you probably see these categories more because they're relatively easier to track because you can control the amount and the route of administration. On the other hand, endogenously released vesicle tracking is much more difficult because of the signal to noise ratio that are difficult to achieve. Yeah, but let's go on. So how do you track them? So essentially you do it by imaging, then this could be done by dissection, or you can actually do quantitative PCR to support your findings, or you can use multiplex method and essentially followed by validation through orthogonal analysis. And by that, I meant using different independent techniques to measure the same attributes of a sample. This is quite important if you're trying to convince people that what you're seeing, especially in vivo, is true. Now, this is one really good example for vesicle tracking over long distances. And this is by using dye. This is a really good work by Vincent Haynes, I hope I pronounced his last name right, where they use membrite to label the vesicles, and they were able to visualize and track circulating vesicles under zebrafish called a plexus. And this is another thing that they witnessed where there's a rapid uptake of the label vesicles by endothelial cells, as well as peripheral blood mononuclear cells. Yeah, see, really nice images, eh? Then moving forward, there's also long distance tracking of vesicle by using contrast agent. This is a nice work by Huang back in 2015, where they used technetium HMPAO mimetic vesicles, and they were able to see this very nice spec CT image to track where the vesicles are going inside the animal's body. So you can see that the resolution achieved by this is really, really high. Another way to track them over long distance is by using reporters. And this is one of the first studies that used bioluminescence. This is Gaussian luciferase that the molecule conjugated to lactoherin. And they were able to track the biodistribution of intravenously injected B16 BLC labeled vesicles. And this is a work that I did back in 2014, where we used Gaussian luciferase that's membrane bound through transmembrane domain of PDGF receptor, and we were able to actually see where they accumulate is including the spleen, the liver, and we further were able to conjugate them with fluorescent dyes to see and validate where they are located. And in 2020, there's another group that published this thermal-loop molecular conjugated CD63, similarly to track where the vesicles are going. And this is another really good one, the ubiquitously expressed CDC3 conjugated to pH lowering to track the interorgan vesicle communication in zebrafish. And I want to mention this one. This one is actually one of the very few, to my knowledge, endogenously released vesicle tracking, where they use nanoluciferase molecularly conjugated to CD63, and it is under tamoxifen inducible cardiac vesicle. So they were able to track where cardiac release vesicles are going inside the body, and they were actually able to quantify and did biodistribution analysis, and they show that those vesicles actually would go to the kidney, lungs, testes, thymus, and such. So this is actually pretty cool. And pretty difficult in my opinion because you're tracking very minute amount of vesicle released by tissues over time. So yeah, kudos to this group as well. And this is done by Kenneth Whitworth's group from Johns Hopkins University. And this is actually through collaboration with us. We're very fortunate to collaborate with them, where they use our reporter and they use suspended XPI P293F cells to generate the label vesicles. And they did a very thorough work to study the biodistribution in both mouse as well as macaque model. And they were comparing the biodistribution either through intravenous or intranasal administration, as well as intrathecal. And they tested different dosages. They were studying the PK, the biodistribution, and saw the uptake by PBMC cells. So this is a pretty thorough study by using multiple models in one paper. So I think it's definitely worth for people to check out if they're interested. And finally, with proper reporter setup, you can actually track both short and long distances at once. And again, this goes back to the Palm Grid reporter that we generated, and as the one that we share with Ken, where we rely on the method of BRET, bioluminescence resonance energy transfer, such that we can actually track vesicles from vesicle resolution, cell tissue resolution to organ and the whole organism. And this is just a quick overview of what we have done. We can actually track where they are in this liver cancer-bearing mice. We can see where the vesicles are located in the organs ex vivo to tissues, to cells, as well as under super resolution, where we can actually resolve two individual vesicle signals. So what's on the horizon? Again, I'm recycling a lot of my jokes. I hope it's funny. I don't know if you guys are as old as I. There used to be a really good electronic music duo. I think it's from France. It's called Daft Punk. If you're old enough, you know what it is. But I really like this album cover because this is an English version. And this is the Japanese version. And if you know a little bit of kanji in Japanese, so the English version says harder, better, faster, stronger. This one goes, work never ends. So they're pretty up to the point. So what's on the horizon? Yeah, our work never ends. So like, are there better reporters out there? I believe, a lot of us, including our own group, we're trying our best to develop better signal, better specificity vesicle reporters to aid with people's study. Are there better dyes that will give you less false positive signals? There are a variety of them. I would say check them out and compare before you use them. Are there better instruments that can provide better signal to noise ratio? And are there better non-labeling? And I think this is a quickly emerging field that I would encourage fellow audience to look into. This could range from Raman spectroscopy, FTIR, and also into third harmonic generation, where they don't require labeling per se, but instead they look at molecular signatures. And I think Stephen Bopar's group is the one that's leading this aspect of the technology into non-label vesicle observation. And finally, like there are a lot of gaps in our knowledge in life of EV or life of pi that to track from the beginning to the end, there are many gaps. And you need to identify whether you want to fill that gap to answer your biological question. So I'm just putting it out there. This is up to your own choice. But if I may, as I mentioned in the beginning of today's talk, this is a conventional vesicle study flow where you apply sufficient dosage to see a function. Then only if you see a function would you be interested in tracking them. I'm hoping that this is a new study flow that people can take a reference of, is that think about a proper way to track them, okay? Don't just choose any way of tracking, but at least identify a way that makes sense to you. And after about 20 years of vesicle, or since its rise to the scientific community, I would say, think about the vesicle subtype that you're looking at. Our group have witnessed that there are actually different size vesicle that can work together. And there are many subtypes of vesicle that people have identified today. So it's important to think about exactly what subpopulation you're looking into. Then think about the dosages. More is not always better because are they physiologically relevant or not? Okay, so this would be very important for both therapy as well as for basic cell biology. Then would you only once you have identified these three components, then you can think more about biodistribution and hopefully how they may translate into function. So the top right is just ChatGPT generated Ghibli style cartoon or animation that you can see for your reference. And finally, as we always want to mention to fellow younger scientists, it's about rigor and reproducibility. And as you can see this image right here, which I created with Gemini, yeah, I'm just trying to tell you, don't always rely on AI nowadays, because look where there are spelling errors, reproducibility. This is completely wrong. So yeah, don't trust it. Trust in yourself. I think this is more real. Okay. Scientific rigor and reproducibility depends on you. And I think this is just some guideline. Try to report how you label your vesicle, or even if it's label free. How did you image them? What are the parameters that you use to label the vesicles? How did you quantify them? What are the controls that you have applied to ensure what you're witnessing is real or not? And how did you validate them by using, say, orthogonal methods? Okay, so I think some other ways that you can report to ensure that other people can repeat your study is really important in this field. And this all depend on you. And if you're careful enough, I can still tell you, don't rely on AI too much. Look at this picture. This Bunsen burner is not even heating the flasks. So yeah, I think that's some thing that we are all going through nowadays. And I think without further ado, I'd like to thank again Ayush as well as Bárbara for co-moderating my talk, and I'll be happy to answer any questions. Thank you.
Thank you so much, Dr. Charles, for your talks. I don't know if anyone has a question that's to Dr. Charles, but I can start. So, in your opinion, which commonly accepted interpretations of EV distribution or uptakes are more likely to be artifacts on the current imaging limitations? For example, I use some controls for my images, but are they really EVs or not?
I see. This is quite a general but difficult question. It would be really great if you can do, say, correlative EM, such that you do fluorescent imaging and then you can do EM. But this is not easy to do. Yeah. So another way is to see whichever way you label them, be it dye or other methods, what would it look like without the vesicles? Yeah, so I see a lot of paper that use non-vesicle. So essentially, their control group are just dyes, and they inject into animals as a control. It's a pretty good effort already, but if you think about it, perhaps a more strict or stringent control would be to use a lipid nanoparticle and label it with the dye and compare with dye label vesicles, and to see whether the signals are real. And it's also important to think about the half-life of the dye. Yeah, as I mentioned, do you think the dye, or do you think the vesicle can last in the body for 100 days? I don't think so. Yeah. So those are things to think about. Yeah.
幫
Yeah.
爺爺上車，爺爺上唔上到啊？咁答。
Yeah, that is actually interesting because I label I put my extracellular vesicles in cell culture for 48 days, and I did an experiment where I label the AVs and I look at them at 6 hours, 12 hours, 24 and 48 hours, and how I know that at 48 hours, I see less extracellular vesicles or less dye. So how they know that the V's are losing the dye, but they are still there, or if are you understanding? I don't know if I'm
Yeah,
too confused.
I can kind of relate to your question. Let me think, eh? Hmm. I would say just from so basically this is more like logical deduction. Like the thing with lipophilic dye is that because they're lipophilic, okay, they may not be readily degraded by cells. And if they are not degraded, most likely they can be recycled. Okay, so since they're lipophilic, so they can be transferred down. And so essentially over time, you might not be tracking the originally labeled vesicles anymore. You may actually be tracking the dyes. Okay, so that is definitely a caveat of any type of labeling. Now, if you use reporter-based labeling, then the temporal resolution might be more matching to the vesicles half-life, because if they're degraded, the proteins are degraded, then chances are the vesicles are also gone. Okay, so that's what I meant, that you have to kind of know. So I'm not saying that the dyes are evil, but instead we need to know the characteristic of the different methods and then apply them accordingly. Right. So I don't know if that answered your question, but you do have to bear in mind that if you track dye or whatever label vesicle over time, think about whether it makes sense or not. Yeah. Because what we had done before is that we actually co-label our vesicle with our reporter as well as the dyes. And what we witnessed is that the signals actually departed. So the dye signal actually went one way and our reporter signal actually went the other way. And this actually go into further detail about detection limit of the different labeling method. And in our hands, we tried one of the lipophilic dye. Remember, it was dye I, and we actually found that the detection limit of the lipophilic dye isn't so great. So our reporter, based on bioluminescence, actually gave much better signal-to-noise ratio. And through that, we decided to use our reporter instead. We were actually hoping that the dye would provide us with a reference. Yeah, but it turned out to be exactly the opposite. Yeah. So those are also things to think about, because if you have a really poor really poor detection limit under the context of biodistribution analysis, you could only detect those signals in the largest organ. And in our experimental setup, the dye actually showed that most of the signal were in the liver or the lungs. But with our reporter, because it has a low detection limit, we actually found that they not only went to the lungs and the liver, they actually go to spleen and other major organs. So then we actually sectioned those, and then we stained them, we imaged it, and then we showed that, yeah, that's actually where they've been to, right? And of course, this is usually followed by another very challenging question, is that after you have witnessed that the signals are there, are they still intact vesicles? I gotta be very honest, this is very, very hard to confirm. Yeah. Yeah, so it's something that I believe could be worked on, but it's just not easy to prove and say that, yeah, the liver vesicles are either intact or not once they have reached their site, right? And that's especially true for in vivo works. But I believe there are many excellent scientists who are working on that right now.
Yeah. We have here a question from Maimona. So, how do you quantify labeling stoichiometric per vesicle, and what are your approaches to avoid over-labeling that could alter EV distribution?
Wow, this is a very good question. This goes into perhaps the labeling efficiency. Still stalking. If I may, but I don't know if he or she's online. I think it goes back to the question you're trying to answer. Yeah, like I think you can use, say, for example, nanocytometry to identify how many percentage of vesicles have you labeled. So that would be, I think, one starting place to see how well your method have labeled your vesicles. Now, does that make sense? Because nanocytometry now can actually detect single particles. And then if your label is fluorescence, then at least you can see how many percentage of them are labeled. So if you actually saturated the system, then you would know. But in our hands, I don't think there's that many 100% labeling method, but that remains to be tried, yeah, with the different dyes as well as other agents that's available out there. Yeah. Sorry, I don't think I answered the question directly, but it does require controls and by using many different methods that you can find. Yeah.
Ayush, you have a question?
Okay.
I had a similar question to the one in the chat box about endogenous EVs, so that covers it. Will you go ahead?
We have another question. Thanks a lot for this very interesting talk. I have a question. The biodistribution of EVs. Most of the studies tracked exogenous EVs, but knowledge on endogenous EV behavior are more limited. According to you, what will be the best model, best approach to study the biodistribution of endogenous EVs from specific tissue, for example?
This is a hard question. Again, sorry, I know I'm like kind of being euphemistic about it. I'm not trying to dodge, but it depends on the question you're interested in. Yeah. So I don't know if your question is coming from a developmental biology perspective or a disease perspective. So it really depends. What would be the best model? I don't want to say it because I think people will kill me if I say this is better than the other, but it goes down to, or at least I could share with you that tracking endogenously released vesicles are more challenging than exogenously administered vesicles. Okay. So keeping that in mind, and I see your question you mentioned about, say, for example, from specific tissue, that may be achievable, such as the one that I mentioned, the one that was published in, let me go back to that, Nature no, sorry, it was Communications Biology. There were lots of, hold on, let me jump a little bit. This one right here. Yes, yeah. So this one, they have the promoter restricted to cardiac cells expression. And I think through that they claim that these are vesicles released by the cardiac cells. So that's potentially one way to do so. And not only that, they use an inducible model. And in a way, it is a control to minimize the background. Yeah. So I don't know if that answered your question sufficiently, but so I would, sorry if I may recommend is to follow through that 7 steps of planning. Identify your question, and then I hope that can provide some general reference during the preparatory stages. Yeah, but it is a difficult question. Yeah, endogenous vesicles is not so easy to track. But again, like it's a part that we're very interested in doing. Yeah.
Going forward, do you think that with the challenges in determining the exact dosage we need, do you think like endogenous labeling is the way forward?
Endogenous labeling is the way
Because
forward.
even with like exogenously administered EVs, there could be a challenge that probably it's not following its natural path. It's just ending up in the lung or the liver, as you mentioned.
Right, this is a great question. So just recently, I think last week, I saw a paper from, I hope I pronounced his name right, Emmanuel Kokuchi from Ohio State University. It was published in Jeff, I remember. And then he was actually reporting that vesicles might actually not travel as far as people imagine, eh? So yeah, so I would recommend people to check out that paper. And then this goes back to your question as to what is the physiologically relevant dosage and how much are they being released? That is not so easy to answer, eh? Because I mean, as you will know, like certain tissues don't proliferate as much. So would it affect their release rate? So yeah, I think we have a lot of work to do. It really so to generate those models to study in vivo takes time. And I believe many people are working on that. It's just that it takes time. So and then but if I may, it goes back to the question you're trying to answer, right? Therapeutic development people, they will try a different dosage, right? Yeah. And then of course they will go further back to how they produce them. Yeah, that's a whole entire different interesting field. So what makes vesicle interesting, in my opinion, is that it covers a range of expertise ranging from biology, chemistry, engineering, all the way down. And then finally, scaling up is also a very important field for vesicles that a lot of people are working on. And what might make it intimidating to newcomers is that it looks so complicated. Yeah. But once you once you kind of categorize them into blocks and think through them gradually or one at a time, and once you make the link, then they start to make a bit more sense. Yeah. So Ayush, I'm sorry, I don't know if I answered your question directly, but I would say, yeah, I mean, it would be really cool to see endogenous, but sometimes endogenous, I mean, even cancer within animal models, right? A lot of the studies were done in, or earlier studies were done in new mice. They did not have the immune system, right? So how do you make sense of that, right? So, and then I think people are trying to look into how vesicles may affect the immune system now. Then, wow, that's a whole entire different ball games that people are trying to do. Yeah. So yes, endogenous is important, but require better ways to tackle them. Yeah.
Thank you. Thank you. That was very helpful.
And I also replied to your answer saying thanks for your answer. We try to do it in zebrafish thanks to specific tissue reporter and CDX tag reporter of EV distribution without great success in physiological condition for normal EV.
Oh, I'm sorry to hear that. So my comment is, how do you define not a great success? So sorry, I don't know the details, but here's maybe you're trying to refer to, yeah, that maybe it comes down to expression level. Yeah, because we do know that any type of reporter expression might affect the cell. Yeah, and we have also witnessed that sometimes expression of certain moiety would affect the vesicle release rate. Right. So that's also something to keep in mind. Yeah. So sorry, Anis, I'm kind of giving you indirect answer or response to your comment. Yeah, I hope that helps a little. So think about what's happening to your cells and whether your donor cells or cells of interest are doing A-okay. Yeah.
I think we have time for one more question. So we have here a question. Many thanks for the presentation. I have a question regarding EV characterization by NTA. Would labeling extracellular vesicles with fluorescence dye improve detection sensitivity? Considering that NTA tracks Brownian motion of particles, I wonder whether labeling would make a notable difference in detection sensitivity when using fluorescence NTA mold.
Okay, that's a good question. Let me see. You're characterizing will labeling improve detection sensitivity? In our experience, not too much in our own hands, not too much, because after all, like it relies on the light scattering, right? Yeah, so in our hands, not too much, but if you use a shorter wavelength laser, you would typically get a better resolution in the smaller size vesicles. Yeah. So, and NTA is not the only method. There are many other methods that's out there that you could also try. So I would say, if I may, NTA is only one of the many methods that you use to characterize your vesicles. So don't be too overwhelmed by it. Yeah. Because there's also zeta view. Again, I have no conflict of interest, so I'm just telling you the ones that I know that's out there. Yeah. And then I think zeta view actually also measures zeta potential. Yeah. So keep those in mind. But if you're using fluorescent dyes especially, yeah, do be careful. Do be aware of dye residues. Yeah, because we definitely witnessed that. Yeah. And there have been reports that they do form micelle, and people actually show that by FACS analysis. Yeah, there was some quite excellent work being done on those. So I do highly recommend you to check those papers out.
So actually, we have here one more question. So how reproducible are NTA measurements? Can you comment on this, please?
Okay, I'll try, Selena. How reproducible? This is like asking how you image your cells. So NTA, again, is imaging based. It depends on how you set your parameters consistently. Yeah, so as if I pronounce your name, Ozge, that its Brownian motion is temperature dependent. It also depends on the concentration. And believe it or not, like it also depends on the size of your vesicle. When it's too big, you don't get very good NTA. Yeah, because the light scattering from the bigger ones will actually cover it, right? So it's important to understand how NTA works. So from there, you're asking about reproducibility. So that would mean that you need to make sure that your image capturing parameters are set consistently between samples. Their concentrations should be relatively the same. That would require a lot of optimization. And it also depends on how you analyze it. So it depends on how you set your threshold. After all those criteria are met, then you should have more reproducible data from NTA. So that's the best answer that I can give you. So there is no one perfect method. It's sort of like there are so many cars out there. Which one is the best car? Then I would say that are you good at driving those cars, right? So it kind of, you have to balance a little bit here and there. And that's what makes vesicle study fun. You got to know the instruments, you got to know the basic principles, you got to handle your samples well, ta-da-da, and then combine all of them to do the best work you possibly can. And then your PI might come and tell you that, oh, this is crap. And then you got to go back to see what's going on. At least that's what I try.
Thank you. So I think it's time to end. And I'd like to thank everyone for joining this session and for the great questions and discussion. A special thanks to Dr. Charles Lay for his outstanding presentation and for sharing such valuable insights into the challenges and advances in EV imaging. It's been truly inspiring. So thank you so much. Thank you, everyone, for joining this session. And you can see this presentation on YouTube.
Thank you very much, guys. Thank you for co-hosting me. Have a great day.